I am using a model of cultured neurons. So neurons cultured on the glass cover slips. And those neurons come from rats. So embryonic rats, not too long before birth. And we isolate this region of the brain called the hippocampus. So the full name would be embryonic hippocampal neuronal culture from rats. And the reason we use this model is because it's a classical model that has been devised by Gary Bunker at the end of the 1970s. Mm-hmm. And that is one of the most detailed models because you can get beautiful neurons that are extremely sparse. So you can really know, you find an axon and you can follow it all the way back to the cell body, and you can really do subcellular work. And so this is the primary reason why we use this model is that if you take a brain or a mixed culture, you know, it's super dense and the cells are all over each other. So if you make a simple staining, you don't know what you're looking at. You know, is this dendrite belonging to that neuron or that neuron? And here you can really spread them and have them very sparse. So you see exactly with your microscope, you know, where are the various compartments and make live cell imaging. And that's the primary reason. And as I said, for me, these are the most beautiful cells. So this I would say the aesthetic aspect is also important.
I also work with primary hippocampal cultures, and indeed they're fascinating. I assume you don't have only neurons in your culture, but also astrocytes, the supporting cells?
So we use this, I mentioned Gary Bunker, and I think the type of culture is called Bunker type because he pioneered the fact that we don't culture the glias and the neurons together, but we culture the glias on the cover slips and the neurons on another cover slip, and then we make a kind of sandwich. So we can have a very low density of neurons on the cover slip with the neurons, but the astrocytes on the facing cover slip are feeding them. So we can, we try to minimize the amount of cover slips we have on the cover slip itself, but this is a sandwich co-culture.
Yeah, we had this exact culture also, but due to the restrictions that are coming with animal experiments, we had to stop them. But this is completely other topic right now.
This is one of the reasons we are interested in these human pluripotent stem cells also, because they are easier to use in that context.
Yeah, but they're still not at all that well developed, but we had a podcast on the topic. Please check it out with Pata Piers Afero. Yeah. So we had a brief discussion while we were walking towards our institute and talking about why it's important to have this culture as a model and what is the relevance for the physiology. Maybe if you can elaborate this.
Yeah, I mentioned that because that's something that's very important. And when you are doing cell biology of neurons, you always have these questions from everyone basically, but neuroscientists in particular. Okay, but these are very artificial cells on glass. You know, is it really relevant? Does it really happen in a real brain, in a real animal? So when will you start doing that on slices or on in vivo and things like that? And I think the logical or the political answer would be, we want to do that. It's very difficult, and actually not. We don't plan to do that. And now I'm more like fighting back a bit and saying, no, we are using a very artificial model. But this is the essence of what a reductionist approach is and a modeling approach is. So I usually answer now, I know a lot of things that happen in slices that don't happen in my cultures. I don't know a single thing in my culture that doesn't happen in the slices. So we are basically simplifying and purifying the process. And what remains in those very artificial cells on cover slips is likely to be very important, because even something as traumatizing and artificial like putting them on glass doesn't remove those very fundamental mechanisms we are looking at. So I try to turn the tables on the argument and say, yes, we are doing something very different, but this is basically the core of what is important at the cellular level that remains.